So let's start. We will start with a module 4.1 part 2. And then I'm going to post the next part, those slides as well. So although we do have a part 1, 2, and 3 for the cancer biology, but altogether there are just like 50 slides approximately. So each day we will do probably just a few of them. So just so that we just meet. So that's about it. So let's start with this. So the first thing that what we're going to talk about, that we already talked about the different junctions in the last part. And here in this part, we are going to go into a little bit of detail in the molecular mechanism that, that is involved in these junctions. So what are these different types? One is the homophilic binding. So homophilic binding means that two adjacent cells, this could be any kind of cells, any tissues, if both of the junctions are the same, they're called the homophilic, homophilic binding. On the other hand, if the receptor, not the receptor, I'm sorry, take it back, the cell adhesion molecules in one cell is different than the other, they're different types, they will be called as a heterophilic binding. Now, when we talk about the cell to matrix, if you have a matrix protein that is binding to the cell, this will be always a heterophilic binding as well. And there is another type of binding, which is where you may have some homophilic cell adhesion molecules in the cell surface on the plasma membrane. And there is a soluble molecule in the extracellular matrix that can aid in binding of the two cells. And we're not going to talk about that. This is particularly rare and very special case. So we're not going to talk about that. So let's see that what these are. So what you have here is, first, we are going to look at these homophilic interactions, and we already talked about the cadherins. And these E-cadherins, so these are epidermal cadherin, and we'll just explain that. And most of them just work in a dimer, like they have just next to each other, and they are interacting with the other one on the other cell. So here is one cell plasma membrane, another cell plasma membrane. They're interacting with each other. And all these dots that you are seeing, these are calcium binding sites. So this is a calcium dependent binding. Both are the same. These are cadherins are classic example of those. The next one is what we called as an Ig superfamily. And what we mean by the Ig superfamily is the immunoglobulin superfamily. Now, please remember that your immunoglobulin or the antibodies that you have, the IgGs, IgAs, IgMs, that is a part of your immune system, those have nothing to do with this. Why we call it as an Ig superfamily? Because their sequences, the repeated sequences of these adhesion molecules do have similarities with those antibodies or the immunoglobulins. And this is a very large superfamily, and that's why we call it a superfamily. Many, many different proteins, hundreds of different proteins. They share a kind of a similar structural basis, but they are not Y-shaped, just like a typical antibody molecule. And they are called the Ig superfamily of the CAMs. And depending upon where they are, there are many different types that we are going to talk about. So again, if they're both the same, they're interacting, they are homophilic interactions. Now, when you go to the heterophilic interactions, the first one, what we are looking at is an integrin. And again, integrin is also a good number of integrins are there, at least more than 22 types of integrin. And here, what they have, that they do have an alpha chain and a beta chain. And we're going to talk about that in a little bit detail. So think about right here is, and you have an alpha variable and beta 3 that is making up. That is not important, just one type. But what it is interacting with is a fibronectin. And this is a ECM, or the extracellular matrix protein. So you can see that integrin and fibronectin are different. That's why this is a heterophilic interaction. Interestingly, you can see that the integrins can act with the Ig superfamily as well. So here is the Ig superfamily. So the Ig superfamily, or the CAMs, Ig CAMs, they can form with themselves a homophilic interaction, or they can form a heterophilic interaction with the integrins as well. So this will be falling under the heterophilic interactions. The next type, what you are seeing, is the P-selectin. P from plasma membrane selectin. And these molecules do bind to many types of glycoproteins. These are the sugars and the glycoprotein background, and they bind to these sugars. And this is actually very important into the white blood cell function. And we're going to talk about this one particularly at the very last slide in very detail. The others are kind of a superficial. That's the only very detailed part of today's topic. So these are the different families of the cell adhesion molecules, which short we call them as CAMs. So let's take a look at the cadherins. So we have already seen the cadherins are found in the adherence junctions where they are interacting with the secondary protein or the plaque proteins with your actins. They will call adherents, where if they interact with the intermediate filaments, we call them desmosomes. So depending upon where they are named differently. So you can see that sometimes this is a kind of a little bit confusing. The E-cadherin were first discovered in epithelial cells or epithelial tissue. That's the reason that they are called the epidermal cadherin or epithelial cadherin, although the epidermis is made up of epithelium, but epithelial cadherin. But they're also found in the pre-implantation embryos. Remember that. That's kind of an interesting just what we mean by the pre-implantation embryos, that the fertilization usually occurs in the fallopian tube or the uterine tube, and they will go into the 4-cell, 8-cell stage morula and almost to the blastocyst level before they get implanted. So these morula cells, which are the 4-cell, 8-cell stage, they are sticking to each other, and those sticking of those cells are responsible. This E-cadherin is responsible for sticking those cells together. And also non-neural epithelial tissue, you can find them. Then you have the P-cadherin, and the P-cadherin the name comes from the placental cadherin. That's where they were discovered, and you can see the trophoblast, which is the lining of the uterus. You can just find them there. Also, you can find them in the epidermal tissue, epidermis. Although they are made up of epithelial tissue, you can find the P-cadherin there. And then you have N-cadherin, nervous system, lens, cardiac, and skeletal muscle. This is known as a neural cadherin. So these are just the different types and names, just to take a look at those. And also remember that your desmoglin as well as your desmocolin, both of these are also cadherin family proteins, but they are found in the desmosomes. So these are all responsible for homophilic interactions. So how do you know that there are so many different types of cadherins and how they are homophilic? That's how do you know. So we will come to an experiment to explain that. So let's take a look at the structure of the cadherin. They are about 750 amino acid. It's a glycoprotein, highly glycosylated. And what they have, that they do have these 1, 2, 3, 4, 5 domains right here. And each of these domains are about 100 amino acid long. And these are kind of a repetitive domain. And out of these repetitive domains, you have about 3 or 4 calcium binding domain. And this calcium binding domain is very important. And the binding of calcium is also needed for the activation of the cadherins. If there is no calcium in the media, the cell will not stick together. And you can demonstrate that experimentally. Then they do have a transmembrane domain. You can see that there is an alpha helical transmembrane domain, and you have a short cytoplasmic domain as well. And that domain will interact with some of the plaque proteins. Here it is, you can see that this could be a catenin, it could be a vinculin, and these plaque proteins will interact with the actin right here. So this is a typical junction, and how a cadherin is at the junction, just one cell on one cell, that has been shown. So if you were just looking at some of these particular ideas, hold on one second. I'm missing one slide right here. Oh, no, that's fine. The next slide. All right. So right here, what we are looking at, this is a typical adherence junction with the cadherin. And you know that adherence junction means that there should be an actin in there. So you can see that here is a cell 1, and here you have a cell 2. So what you are seeing, that here you have the cadherin from one cell, here is you have the cadherin from the other cell. although they are colored differently, but both of them are E-cadherins, both of them. There is no difference in between those, although they are colored different, just for your understanding that they are from the opposite cells. It's kind of interesting. This part is not needed, but just have an overview of the idea that if you don't have this cell 2, if you just only have this cell 1 in isolation, which is not interacting with the cell 2, the cadherins actually of the same cell are going to act with the other one. So this is one cadherin molecule which is interacting with the other one. And when they are acting to the neighboring cadherin, we call them as a cis, C-I-S, cis-acting. So these are the cis-acting cadherins. But when one cadherin will interact with the opposite cadherin from another cell, we call this a trans-acting. We use that term all the time. So cis on the same side, trans across with that. So these are the two formation that what you have. So you can see that you have a monomer of cadherin that is interacting with another cadherin. Here you have one interacting with another. So this is active in a dimeric form. Then what you have the transmembrane domain that you talked about. And then there are a number of other proteins which are the connecting proteins. And these connecting proteins could be beta-catenin, alpha-catenin, and here is one which is called a ZO1, which essentially stitch multiple filamentous actin, which is added with alpha-catenin. They just can stitch these together. On the other hand, on the other side, you are seeing that there is a separate set of proteins. You have this vinculin, then you have this VASP, which is a vasodilator stimulated phosphoprotein. What we mean by that, if you just do have a particular signal, or even when you are working out and you have a condition which is a vasodilation, means your capillaries actually enlarge in diameter or just become larger. At that time, this is activated. But again, these are not needed. But just to show you guys that right here, you have a P120 catenin, alpha catenin, beta catenin, vinculin, or lots of these different proteins. But ultimately, all of these proteins are interacting with the actin, and that's your adherence junction. Now, the important thing is that although you can see that here you have one set of protein, here you have another set of protein, there is no reason to think that this should be different. They could be different, but this cell may have exactly the identical proteins of this cell as well. So both the sides may have the exact same type of plaque proteins, which is connecting with the actin. So that is a very important idea. So this is a typical adherence junction of how the E-cadherin connects the two cells. So the next thing that we talked about, that this is a homophilic interaction. So a typical type, and that was I was just looking for. So E-cadherin only binds with E-cadherin. Same thing for the P-cadherin and N-cadherin. So EP or PN or NE, they will not interact. And how it was actually determined that the scientists actually isolated some cells which are called L cells. These are kind of a enteroendocrine cells. They secrete some of the hormones in the intestine. That is not needed. But these L cells are found in the intestine. So they isolated some of these L cells from the intestine. And why they basically isolated those, these are some of the cells that do not express cadherin. So there is no endogenous cadherin in those cells. So what they did, that they transfected the genes of specifically for E-cadherin, then P-cadherin, as well as the N-cadherin to those cells. So those cells now expressing the E, P, as well as N-cadherin, they mixed all these cells. And of course, each of them will have a specific antibody to them, and you can just isolate them or visualize them under the microscope. But what they found, that this is the experiment by which they found that only the E-cadherins will interact with each other, only the N-cadherins and only the P-cadherins will interact each other. So this was the experiment which basically showed that cadherins always bind to the same cognate partner, which is a homophilic interaction. All right, so here is also another cell right here. You can see that these are some of the MDCK cells. These are some dog kidney cells, which are expressing a GFP tagged cadherin. And you can easily see that they are just over the period of time, they are interacting with each other. And once the cadherin are zipping together, you can see that just right at the junction where you can see that the cadherin is really fluorescing. So that just shows that GFP cadherin, the response that's being in the junction of these cells. All right, so that's your homophilic interaction of cadherin. So the next one, what we are looking at is the homophilic interaction of the Ig superfamily, immunoglobulin-like superfamily. So here, the immunoglobulin-like superfamily, again, it's a large, very, very large protein of many different types, and also calcium-independent homophilic interaction. Remember that this is very different. Cadherins require calcium, where immunoglobulin-like superfamily CAMs don't require calcium. And also, they do the homophilic interactions, means they can act with each other, similar type, but they also do have a heterophilic interactions with the integrin that we have seen. So what are the different types? Say, for example, NCAMs, they are in the nerve cell, which are called the neural CAMs. And then ICAMs, they are found in endothelial cells. They're not called ECAM. So I stands for intercellular CAMs. So that's how it's named. That's why it's ICAM, intercellular CAMs. So what are the different types? You can see that they do have these repetitive structures, and these the red structures that you are seeing, that these are the disulfide bridges. So disulfide bridges make a very typical type of structures. That this is a schematic cartoon, of course. But what you can see here, that some of them may have a long cytoplasmic tail, some of them a short cytoplasmic tail. Some of them can be anchored with this GPI anchor in the lipid or the plasma membrane, doesn't even have a transmembrane domain and a cytoplasmic tail either. On the other hand, there are some Ig superfamily CAMs which are completely soluble. That means they are in the extracellular space, and those are responsible for binding or to the cells as well, to the extracellular matrix. So all different types of immunoglobulin superfamily CAMs are there, and these are some of the things that is important. So what we are looking at is the integrins, and we're going to look at some of the heterophilic interactions as well. So already we have told that right here you have the extracellular matrix, the picture from the previous slide set, and the extracellular matrix ECM interact with this integrin. So that is why this is very important. So the integrin do have a very typical structure. So what you have is an alpha chain, and this alpha chain is in two parts, and these two parts are connected with a disulfide bridge right here. And then you have a transmembrane domain and a short cytoplasmic tail. The most important part that what you have multiple calcium or magnesium ion binding sites. So for the functioning of the integrins, you absolutely need either calcium or magnesium. You do not need both. Just any of them will activate depending upon which tissue they are. There are some integrins which are magnesium dependent. There are some integrins which are calcium dependent. So it depends what they are. Then you have a beta chain. So this is a heterodimer. You can see that because these two dimers are not the same, they're a little bit different. That's why we call it as a heterodimer. And you have a beta chain. That beta chain do have a characteristic region, which is a cysteine amino acid rich. Then you have a transmembrane domain, and then you have the cytoplasmic domain. So the beta subunit bind to some of these plaque proteins or the connecting proteins, which are talin and alpha-actinin. So this is a kind of a quiz question that you can think about, that your talin or alpha-actinin, which is the interconnecting protein, which binds it with the intermediate filament or the actin, they are the intermediate. These proteins or the plaque proteins are present or interact with the beta chain, not the alpha chain. So this is something even important. So that's your alpha-actinin and beta-talin or talin. So here you are looking at a number of integrin. There are more than 22 different types. You don't have to memorize all of them. I'm just pointing out that what you kind of understand from here, there are many different types of subunit composition. And what the take-home message, I will just point out some of the ones which are very specific, but what it shows that you have the combination of different types of alpha subunit and beta subunit, and this permutation and combination gives them specificity to different types of primary cellular distribution and the ligands. So just one example that is very important, that just make sure that these are the just only 2 or 3 that is important for your quiz or exam. If you have a subunit composition of alpha 1, beta 1, many different type of cell express that, and they mainly bind to collagen. And this is collagen is in the extra ECM, or this is in the extracellular matrix. So alpha 1 and a beta 1. Now, if you change this alpha 1 to alpha 2, so now you have alpha 2 and beta 1, also found in many types of cell, it still binds collagen. But see that what is added is the laminin. Remember that. So if you remember that from long back, we talked about this nuclear lamina, where we had a confusion with the lamine and the laminin. In the nuclear lamina, the protein that you have is a lamine. This laminin is present in your extracellular matrix. So the take-home message from this is that just changing this increases the specificity of the integrin. It still binds collagen, but it now can still bind to laminin. So just these first 2 examples, that just remember that. And then another specific one, remember that right here, because we are going to discuss this in detail. If you have an alpha L, means alpha long, and beta 2, if you have this configuration of the integrin, they are found on ICAM1 and ICAM2. And these are intercellular CAM1, intercellular CAM2s. So they are found on the T lymphocyte. This is just not very inclusive, but they are also found in monocyte and neutrophils as well, but many different types. So you can see that right here, although it's written as monocytes, but these are present in many different types of cells as well. But again, just if you remember that alpha L beta 2 binds to ICAM1 and ICAM2, because we will discuss this in a little bit more detail. So let's do the first clicker question. So our first clicker question will be, which of the following is needed for the binding competency of E? Oh, there is a spelling mistake in there. E-cadherin. Good, everyone? I'm stopping it now then. Stopping it now. All right, so 50% calcium is the correct answer. So if you just go back, you can easily see that if you just right here, if you look here, see that this is 3,4 calcium binding domain. So cadherins are only calcium dependent. Only calcium dependent. That's very important. On the other hand, if you see right here, your immunoglobulin superfamily is calcium independent, and then you have the integrin, which is calcium or magnesium dependent. So about 31% said A and B, which is the either calcium or magnesium, but just remember that for E-cadherin, it's calcium only. For the Ig superfamily, nothing is needed. And for your integrins, calcium or magnesium, which is either A or B, could be correct. So remember that. I will just put all these clicker questions, but remember that, look at this, that this can be changed in your exam question. This could be replaced by your, with your integrin, or this could be replaced with the Ig superfamily CAM, and then choose the answer correctly right here along with that as well. So just make sure that you just take a note of the clicker questions. I am not posting the questions because I just answered this to a couple of people that when I was evaluated by the peer evaluation, most of the people told me not to post the questions and the answer of these clicker questions, because you guys need to work a little bit harder. So I'm just following their recommendation. They also told me not to post any of the transcript, but I thought that, okay, all right, I just at this point, I'm not going to change that. Anyway, let's go back to the material. So here, what we are looking at, the cells can actually regulate the activity of the integrin. And what we are going to see, that integrin is also a signaling molecule. So it is a very versatile molecule. And what we are seeing right here, very interesting, that here you have an integrin and you have a receptor, entirely different type of receptor. And here in this condition, the integrin is in an inactive form. And here you have an extracellular matrix, and you can see that there is no interaction and there is no binding with the extracellular matrix. Now you have an activating signal, and that binds to a particular receptor. And then there is a series of proteins which comes and binds to the integrin, and there will be a conformational change, and the integrin will convert into an active integrin. And at that condition, what we call as this integrin is now adhesion competent. At this point, it can bind to the extracellular matrix. And we are going to see an example of this when we talked about the mechanism, just taking one example. So again, very, very nicely, you can regulate that. On the other hand, you can also do this in a little bit different way. That here, what we are seeing is the deactivation of the integrin. Here you have the extracellular matrix. The integrins are being bound. Here you have the attachment proteins on the actin cytoskeleton, and these are your plaque proteins. And here you have perfect binding. Now what can happen, that if you phosphorylate the serine residues on the beta chains right here, at that time, remember that it's a beta chain. So this is a small thing right here, but this is a good question for your exam. So serine residue on the beta chain, if it is phosphorylated, this whole complex falls apart. So the conformational change will no longer will be able to bind these attachment proteins. At that time, there will be no binding and they will all disperse. So this is basically a process of deregulating or deactivation of integrin. Now, where this actually happens, the very, very nice example is the focal context. So think about that. A cell is a motile cell and it is moving on the substrate. So what is happening at the leading edge, it is lifting up the lamellipodia and is attaching the lamellipodia onto the substrate. At that time, where it is attaching to the substrate, you will have activation of this. So this will go on. On the other hand, as it is moving at the trailing edge of the cell, which is being lifting, that part is being lifted up, and there you will have a breakage of that. So that is going on the trailing edge of a moving cell. This is happening as the leading edge of a moving cell. So breaking and making of the focal context, very, very nice example of a motile cell. Also, this is important in the mitosis. So remember that when you have, following your telophase, you have cytokinesis, all these cells, or the two cells, which are the daughter cells, are elongating, and they were earlier attached to the neighboring cell. But when they divided into two, they are actually going away or elongating in such a way, if they are attached still strongly to the neighboring cell, they will not be able to elongate. So you need to break that interaction so it happens to that as well. Unfortunately, what happens in the tumor cell, just not only the tumor cell, the more correct word will be a malignant tumor cell or a cancer tumor cell. Tumors could be benign, and there it doesn't happen. Usually it's a malignant tumor or cancer tumor cell. This interaction happens. So this phosphorylation breaks those, and that's the reason these tumor cells will move away from the origin of the tumor, and then they will migrate to different tissues. And you all know that what is that is called? That is called a metastasis. And that's the reason that the tumor lose the integrity, because these attachment in between the cells in the tumor is gone in the cancer cells. So this is the regulation of your integrin activity. All right, so let's take a look at the next one. So here what we are looking at, that we are looking at the model for the integrin activation. So this is a molecular model right here. And first, let's take a look at the cartoon. So what happens that when integrin is inactive, you have these two domains. You have this beta and the alpha domains. And they do have this typical structure where they're kind of bent over the plasma membrane. Right here is the plasma membrane, they're kind of bent over. So what you have that you can see that chain, and right here, this is a typical structure which is known as a beta propeller. And now, well, this is a term that is used by the crystallographer or the people who actually do the structural biology. And the reason that they are called this as a beta propeller, because just it looks like that. I can show you some nicer picture, that you have some domains like those repetitive domains. Think about that. These are like the blades or fans of a propeller. They are similar like this around a focal point or around an origin. They are very, very homogeneous in that distribution. And if you see that these thick bands, these are all beta sheets. And that's the reason that with the beta sheets, they give a propeller-like appearance. And that's why this fold, this is a very typical fold of your protein, three-dimensional protein structure, which is called a beta propeller. And here you can see that the beta alpha domain. And right here, this is your beta A domain, which is the top portion right here, which is right here, which is the beta A domain. And the next portion is right here, that is your rest part. So when integrin is activated, as if what you think that they just kind of interact, this beta A domain and the beta propeller interact with each other. And in this condition, what they do, they are pushed apart. So if you carefully look at that, in an inactive form, this beta alpha are close together. The transmembrane domains are very close together. And in this condition, they are kind of bent over, and your beta propeller and the beta A domain do not interact. This is your inactive form. But what happens that the integrin, when it receives a signal, there are some conformational change, and these two transmembrane domains are pushed apart. In that, these two interact and they kind of stand up. At this point, the integrin is binding competent. So just remember that how it activates. So this is the overall process. So here what you can see that integrins also, this is it's a very universal molecule. They can serve as a signaling molecule themselves as well. So make sure you carefully understand that what is needed from this slide and what is not needed. Remember that we are talking about here the cellular interaction, but we are not talking about signal transduction, which we did in the last part. So I will not ask you these specific pathways, which is what after this. You just have to take home message from here that integrin, how it can activate the signaling pathway. So what you are seeing that an active integrin, when binds to a ligand right here, it can activate several pathways. And this one you need to remember that it also can activate a pathway which is a receptor tyrosine kinase activated pathway. This pathway, this names you again do not have to remember, but if you remember from your previous chapter, see that once the receptor tyrosine kinase is activated, it will, the GRB2 will be incorporated, then SOS will be incorporated. This is the guanine exchange factor. RAS will be activated, which will again activate RAF, MEK kinase to MAP kinase, which will lead to the different types of cell cycle as well as gene transcription. So this same pathway, which is activated by the receptor tyrosine kinase, can be activated. The integrin can activate it by the same pathway. And here you can see that this is a PI3K pathway. We did not talk about that. And then there are some RAC-RO dependent pathway that can be activated as well. So just only to your take-home message from here, just remember that integrin can activate several different pathways, details not needed. The only thing is needed that it can also activate the same pathway which is activated by the receptor tyrosine kinase or RTK pathway, because you have learned that. So these two can be similar. That's it. But individual steps are not required. All right. Any questions on that? Anyone? Okay. All right. So after that, we are looking at two interactions. One of these interaction is a selectin, and this P-selectin with the sugars present on some of the glycoprotein, and we're going to talk about this, and also the integrin with the Ig superfamily CAMs, mainly not NCAMs, but the ICAMs. Both of these heterophilic interactions are important for the white blood cell function. Not all functions, but some specific type of functions, and we are going to talk about this in the detail, these two heterophilic interactions. So here we will spend a good amount of time in this slide because this is a lot of information in here, and make sure that this is one of the important slides because a lot of question can be asked from this. So let's first let me give you an overview of this, because kind of this particular part is cell biology, a little bit of anatomy and physiology, a little bit of immunology, everything all together. So just think about that right here, you have a connective tissue. And this connective tissue, just think about that this is the part of your skin, which is the dermis part of your skin, which is a connective tissue, and it does have blood capillaries. So right here, what you are seeing, let me just get a pen right here. Okay, so right here you have a blood capillary, and this blood capillary does have these cells. This is what you are seeing right here. So these cells right here are these cells. And right here, suppose this is your skin surface and you have a wound right here, somewhere, not that close, right here. You have a pathogen invasion or wound in this area. So what will happen that when you have this a pathogen invaded, suppose you have a bacterial infection or a viral infection, or you have a cut or a wound right here on your tissue close to a blood vessel. And remember that this is just a capillary, a very fine capillary. And when this happens, the cells in this area, they will release what is called this inflammatory response. And these are the cytokines, the different cytokines, the chemokines. And this is like you do not have to remember the names, but interleukin 1, IL-13, IL-17s. And these different inflammatory response cytokines will act on these cells right here, all these endothelial cells. And these are the endothelial cells on one side. The top is not shown. So what you are seeing right here on these endothelial cells, these are the white blood cells that you are seeing inside the capillary. The top portion of the capillary is not shown. So what is happening right here? Let's take a look. So what you are seeing right here is an endothelial cell, which is lining the blood capillary right here. And this contains a vesicle which contains the P-selectin. And this P-selectin is the molecule attachment protein, and that is not on the cellular surface. It's inside the cell in the vesicles, but they're already preformed. They're just right there. And on the cell surface, you have this ICAM, the intercellular cell adhesion molecule. That could be ICAM1, ICAM2. So in the normal condition, you are seeing that there is a white blood cell, and they are not all different white blood cell. Usually, if you have a tissue injury, the main cells which will be called in to fight the problem here, tissue injury or invasion, will be the neutrophil and the monocytes. Now here, this is not needed, but the monocytes, when they are inside the blood vessel, they are called the monocytes. And we're going to see that these, if they're monocytes, they're going to leave the blood vessel and come into the tissue space. From here, they will come into the tissue space that is being shown here. They will be called as macrophages at that time. So the monocytes circulate in the blood for about 8 hours. And then from the blood, they will move into the tissue space if needed, and they will be called as the macrophages. And also the B cells and the T cells do have this same condition that can lead to this tissue space, and they will come out of the blood capillary into going into the tissue space. So what you have on the leukocyte, this leukocyte means any of these. So on the surface of them, they have something which is called a platelet activating factor receptor, PAF receptor, platelet activating factor receptor. Then the yellow one is a selectin ligand specific carbohydrate, and this is a mucin-like CAM. Mucin is a glycoprotein, which is not written here, but that will bind to the selectin, and we're going to see that. So this is a selectin ligand. And also they have this alpha L beta 2 integrin, and you can see that this is the bent form that we talked about. So this integrin is in inactive form on this leukocyte. So what is happening that when you have these endothelial cells in a normal condition, you don't have any tissue injury, you do not have any pathogen invasion, all these leukocytes are rolling past, or they're moving with the blood flow through the capillary in an extremely fast pace. I will actually post a video, you can see that, extremely fast pace they're moving. But what happened once you have this tissue injury, at that time, these vesicles containing the P-selectin, they will be trafficked on the cell surface of these endothelial cells. Now you have these P-selectins on the cell surface. This will bind to this selectin ligand, which is the mucin-like CAM. So you can see this. So when this happens, it will capture the leukocyte, and the leukocyte will kind of roll onto the surface of this. So they will kind of roll. So why this is happening? Because this interaction in between the P-selectin and the selectin ligand is not a very high affinity. So the shear force of the blood that is pushing these will allow it to break and form, break and form, and break and form. So they're kind of just moving along. Slowly though. Earlier it was just not interacting, it was just moving extremely fast. Now they're kind of rolling on the endothelial cells. While they roll, what will happen at that time also, this is a platelet activating factor. This will be expressed on the endothelial cell. So there is a difference. See, the P-selectin was in the vesicle that is trafficked on the cell surface, whereas the platelet activating factor will be a transcription activation due to those cytokines, and that will be expressed on those. And the platelet activating receptor of the blood cell or the leukocyte will bind to that. And at that time, now what is happening? The integrin, this platelet activating factor receptor will undergo a signal. We talked about that it can regulate that, and your integrins will be activated. Now you can see that they are straight. So at that time, it will bind to either ICAM1 or ICAM2. So when this happens, the interaction of the integrins are way more stronger compared to this P-selectin ligand interaction. So this P-selectin and the mucin interactions allowed the cell to roll, whereas here, the integrin and the ICAM interaction will pull the cell very close to the endothelial cell. And when they have a particular area in between the endothelial cell, the platelet activation factor will also induce the motility of these cells, and it will squeeze through this area into the tissue space. So basically what will happen right here, if we just see that these cells, they will squeeze through here and come into the tissue space. They will squeeze through here and come into the tissue space. All these cells will be now there near this injury site fighting the infection. And this particular process of moving out of these blood, the capillary or the blood vessel into the tissue space is also, this crawling out is also known as diapedesis. That's, if you take anatomy physiology course, you will understand that. So this is the whole process. Just make sure that you understand this process. It may take a little time, so read the material as well. Just listen it to a couple of times. You should be all right with this. So that's pretty much overview of the whole thing. That's the 4.1. So we talked about the tight junctions right here, which occluding junctions separate the proteins in the basolateral and the apical surface. We call the adhesion belts or the adherence junction. When they interact with the actin, they are called the adhesion belts or adherence. When they interact with the intermediate filaments, we call them desmosomes. And we have the gap junctions or the communicating junctions. Then if we have the basal lamina or extracellular matrix interactions, we call them, if they're anchoring, we call this hemidesmosomes. And these focal contacts, which are important in movement or the motility of the cell. That's important. Then we can talk about the integrin with the, again, these are non-junctional adhesions, which is the extracellular matrix or lamina with the integrin. You can have the cadherin, which is a homophilic interactions. Ig-like CAM, they can also have homophilic interactions. Integrin Ig, heterophilic interactions. And we just saw the selectin and mucin-like CAM. These were the two heterophilic interactions we just saw. So that's pretty much all of the chapter, I guess. And let's do the last quiz. So right here, which of the following activates the integrins on the white blood cells in response to a tissue injury or inflammation? P-selectin, mucin-like CAM, platelet activation factor, platelet activation factor receptor. $15 seconds. Stopping it now. Good, everyone? Okay. All right. So here is some confusion. And the reason I put this question is it is a little bit confusing. And there could be two answers. So, and I will explain that why it is. And the question is not complete because it is a little bit tricky. And this type of question, once asked, you know, that I usually throw out these questions. So so the question is that this could be correct answer, but this could be correct as well. So I should have given a CD where both CD is correct would have been proper. And I will just explain that why both of them technically could be correct. So I'll just go back to that. All right, so let's take a look here. All right, so what is happening once you have the inflammatory response, the endothelial cell is expressing the platelet activation factor. So now what happens, the platelet activation factor is now bound to the platelet activation factor receptor. So you can always argue that once the platelet activation factor is bound to the receptor, now the receptor is responsible for activating the integrin. If there, on the other hand, so you can think about it that the receptor is changing something and that will activate the integrin. On the other hand, you can say that the platelet activation factor, if it is not bound to the receptor, in the native state, this receptor is not going to activate the integrin. So are both responsible for that? And that's where the confusion comes in. So that's where these type of questions, sometimes if I ask, just make sure that you always ask back, because sometimes I just put those questions, and today I purposefully put this question because these are some of the confusing questions that sometimes will confuse you. And if this happens, by the end of the day, I will open up the quiz and just always ask, because this is actually the factor which, unless activates the receptor, this will not be able to activate the integrin. So if I'm just given the first choice, I will always choose the platelet activation factor. But what I'm saying that someone can argue that if this receptor was not present, then this integrin will not be able to activate. So just the presence of PAF is not enough. So why that is not the answer? And in these cases, it is a little bit confusing. And these are some of the questions that I throw away because you can think about this way. So a proper question, probably, if I just think about this, without tricking, that the activation of the platelet activation factor, so this is how it is, platelet PAF receptor activated by the PAF should be the correct answer. So you can just think about that. I'm just trying to just rewrite that question like this. So platelet activation factor activated PAF receptor. And I will just probably take this one completely out. Platelet activation factor activated receptor. If you just only have, on the other hand, just only PAF, or if you just only have the PAF receptor, you don't have these two choices. PAF receptor, you're fine. You will always choose that. On the other hand, if you don't have this choice, or if you don't have this choice, then you can choose the PAF, that's the answer. You do not have these choices, that's the choice. But if I give you both of these choices, that's kind of confusing. So when I ask the question, I will not ask this question. And I will explain this particular question in here, because what I see, some of the people just get confused by this, and that is very normal. So that's all good. All right, so I think that's pretty much it. Any questions, folks? Anyone? No? Someone can talk to me.
We can hear you, but I think it was pretty clear.
It's good? Everybody good? Okay. And
Dr. Q, I have a question.
Yeah.
Several of us have noticed that our exam 2 and exam 3 grades have disappeared from Canvas, and I was just wondering.
Yes. So the thing is that the reason for that, yeah, I absolutely know. So what happened is that someone is taking the exam 3 today, and there is one person, there was a complete different scenario, who has not taken the exam 2. He will be taking the exam tomorrow. So that's the reason that it disappeared. So it's not, I will just post that. So it is not a problem. So those exams are only open to them. And once they finish that one, I will open it up. So you will see that today after 9 p.m., your exam 3 will be completely open, and you will be able to see your responses and answer. I will keep them open. So there is no window or anything like that. You guys are already having enough. So you will be able to take a look at them. And after that, if you see that some of the questions which you have questions, you just send me your queries, and it will be all good. And your exam 2 grades will be available again, same grade, no worries, will be after Wednesday after 9 p.m. So good, everyone?
Great, thank you.
Yeah. Anyone else? Anything can think about? Let's see what we have right here. Okay, all right. So someone told that also, when will the course evaluation be up for this course? All of my other courses are due next week, but all the main page said is contact the instructor when it will be posted. This is a kind of interesting thing. So what happens that this semester, there is a new course evaluation, which is called as an eval kit. Did anyone of you got that new communication regarding that?
We did not.
So that's kind of so let me just go back, and I don't know when it is. So what they told that they are doing this eval kit for the first time. So when you guys did your other course evaluations at that time, did you guys use the old system, or did you guys use the new system?
I just went to the course evaluation like I've always had, so it had to be the old system.
Okay, so you could do that for other courses, right?
Yes.
Okay, so what I will do that I will just not just mess with that, with the new system, because we were told to deactivate the old system so that the new one, which is the eval kit, comes in. So what I will do that I will just go right now, right over here, and I will activate all the course evaluations. So you can use the old system to just go in there. And I highly urge, please go in there and write what you feel about the course. It doesn't have to be nice. Remember that. It is completely anonymous, doesn't have to be nice. What you exactly felt, and your honest review is the most important part. And again, remember that this will be open all the way up to the last day of your grades are posted. So I will post your grade probably just a day or two after your final exam. So you will have 5-6 days even after your final grades are posted. So you have plenty of time to do that. I'm not going to close it early. Remember that. I'm going to keep that open all the way after. Many people just close that before the final grade, but I'm not going to do that. So you have plenty of time. Good?